Hello, everyone. Welcome to Veryst Engineering webinar. We're going to give people a couple minutes to get into the meeting here, but hopefully you all can hear me. So we're going to get started in 1 minute. But if you're interested and you know you have questions now, you can go ahead and throw any questions into either the chat and the Q&A. Matt and I will address those as they come, as well as have some time at the end for some questions from the audience. So please feel free to throw those in there. All right, we'll get started. We're a little bit after the start of the hour here. Thank you very much for attending this webinar on multiphysics simulation and specialized testing and combination product design and verification. This is part of Veryst Engineering's free monthly webinar series where we talk about our capabilities and the type of projects that we work on for our clients. A little bit about us here at Veryst, and the two people giving the talk. So on the left, you can see Matt Hancock, my partner, who's waving there for you. His background is in fluid mechanics and microfluidics. He's got his MIT from PhD, postdoc at MIT and the Harvard Weiss Institute, and has been here at Veryst working on fluid mechanics problems, focusing on microfluidics for a good part of his career now. Over on the right, you can see me. My background is material modeling, testing, and solid mechanics. I got my PhD from Brown University in solid mechanics, and I spend my time fairly evenly split between working on projects that are just doing finite element analysis, FEA, for our clients, a lot of projects working on material testing, and then in between that material model calibration, taking test data and putting it into our finite element models to help our clients answer the questions that they have about their product or process performance. So overall, Veryst Engineering is a group of subject matter experts with varying backgrounds that overlap, and we work with our clients to help them solve their most challenging problems to accelerate product development or manufacturing and deliver those better products to their clients. We do this by using our expertise in mechanics, material science, chemical engineering, EM, failure analysis, and material testing to inform our clients. And we do this, we like to say we do this with academic level expertise, but on an industrial time frame. Over on the right, you can see kind of a wide range of the subject areas that we work in, from polymer and material testing and materials engineering, down to heat transfer, multiphysics simulation, electrochemistry and reacting flow, fluid mechanics, and solid mechanics. We put all that together in multiple projects to help our clients, and we've put together a bunch of examples today for you from the combination products industries to talk about how we've solved clients' problems to get their products to market faster. We like to take a hierarchical approach to our problem solving, and we use a wide array of tools to do this. Many times we start with pen and paper and ideation, sitting down with our clients, understanding the problem and how they're trying to solve it, and seeing if we can guide them quickly by getting analytical formulas or some research to help them push the boundaries of their product development and get them moving faster. After that, we typically move into testing and modeling. Depending on the project, we could be working with them on full product tests or just material tests, and we'll usually use that data to inform and develop a model. Again, sometimes these models are kind of analytical models that you can't solve by hand, but you can solve and get relationships out of to predict performance. But sometimes they're full simulations, whether that's solid mechanics, fluid mechanics, or a multiphysics problem to help them predict that. We always try to keep in mind the outcome. What is the client trying to develop? What are their roadblocks? And how can the simulation or that model answer the question and develop that product and help them reduce iterations to get to market faster and increase patient safety. Last, we like to say that we don't just give you some results from our simulation and leave you to interpret them yourselves. When we do these types of analyses, we then interpret those results for you and give you our recommendations based on the data that we have and how to make your product or process perform.
Thanks, Sean. So one of the ways we use our expertise and analysis tools is to test design ideas, to provide feedback on feasibility and optimal design. Early in the design cycle, it's often much faster to test ideas with simulation rather than, say, prototyping, to assess feasibility and performance, discover sensitivities and potential problems, and test interactions with other parts of the system. Simulation helps develop a better first prototype, and that saves time. We've worked on examples where multiphysics simulations can increase a concept's credibility and reduce to practice in silico for IP disclosures, some of which are shown here. We've also worked with groups doing new technology search to assess feasibility to incorporate into their existing systems. We have a great deal of experience modeling the behavior of injection devices with dual chamber cartridges. Work published this summer outlines our work with Merck to model spring-driven dual chamber autoinjectors. Autoinjectors with dual chamber cartridges are single-use self-administrable devices that facilitate automated reconstitution and injection of freeze-dried drug products. Our model that we developed and that we presented in the paper predicts several essential performance requirements, including injection time, stopper trajectories, and other criteria, such as the maximum diluent volume that allowed a certain target performance. Our mathematical model was developed based on Newton's second law of motion, Hooke's law for the linear spring, the ideal gas law, and a custom stopper friction law that we derived from our in-house experiments. The model provided good predictions of injection time compared with experiments, and the model really, the goal was to be used by formulation scientists to rapidly perform a virtual test of different devices and formulations. This reduces experimental time and cost and accelerates the drug and device development. So it turns out that predicting stopper friction from first principles is complicated, and there's a number of reasons for that. There are different physical mechanisms. It's usually a pretty small scale. The thickness of the lubricating layer is submicron. So rather than, for this particular project, rather than start from first principles, we instead experimentally measured the stopper friction at different speeds in our laboratory. We then fit an empirical power law curve to the average stopper friction, and then we were able to incorporate that into the mathematical model. And that gave very good predictions, but it also shortened the time to develop the model. The dual chamber cartridge we modeled is shown here. A spring connects to the primary stopper. The spring actually isn't shown, but it would be coming in from the bottom. Diluent and freeze-dried drug product are kept in sealed chambers, and they're separated by a secondary stopper. The model predicts stopper motion across 4 stages of dual chamber autoinjector operation, and those are reconstitution, priming, and injection. Once the secondary stopper moves past a bypass channel, air and diluent can pass into the drug chamber. And this starts the reconstitution process. Air pressure builds up in the drug chamber, balancing the spring force, and the stoppers come to equilibrium. Drug dissolution and mixing occur in a step that was not modeled, though later I'll discuss how we can use multiphysics simulation to address that step. Once the solution is mixed, The user activates the needle to pierce the seal in the closure cap, and this allows the trapped air in that second chamber to escape, and then the stoppers continue to move. The priming and injection steps follow after that. The model uses drug solution viscosity measured a priori, and it's provided as a model input. The model predicts both injection time and flow rate. So the dual chamber autoinjector model that we developed, it's got inputs related to the device, to the primary package, the formulation, and the release medium. Since the model is reduced order, this is just solving a differential equation in time. It solves within about a minute to simulate how the design changes impact key performance metrics. And these performance metrics would include things like injection time and stopper motion. The predicted stopper trajectories are shown in the left plot through the various stages of operation. Notice the rapid initial motion, and then later on, there's an equilibrium just prior to seal puncture, priming, and injection. One of the important steps in model development is validation, and in this case, we did that with experimental measurements of injection time, and the model did a good job of predicting injection time. So again, the model can be used as a quick check by formulation teams to test things like making sure the stoppers get to the target positions. It can also be used to infer, to predict injection time. But certainly there's other aspects of the reconstitution process that we would need a different type of model, and I'll discuss that in a few slides. So one of the advantages of having a model like this, once it's developed and validated, is that it can be used to probe sensitivities to design, operating, and material parameters. The model predicts that injection time is sensitive to needle gauge and drug solution viscosity. And it also predicts that injection time is roughly proportional to viscosity and the diluent volume. Now, going one step further, for low viscosity drug product solutions, injection time is less sensitive to needle size compared to, say, higher viscosity solutions. Also, the hydrodynamic resistance is low for low viscosity drug product solutions. And so stopper friction, which is independent of that viscosity, it plays a moderating influence. For high viscosity drug product solutions, the needle hydrodynamic resistance dominates stopper friction. And so injection times and flow rates inherit the hydrodynamic resistance's sensitivity to needle size. So to model the dissolution of the lyophilized drug product and mixing of the dissolved drug product, we developed a COMSOL multiphysics model with coupled flow, species transport, and the population balance equation. We used a typical autoinjector chamber geometry, and we predicted the transient dissolution and solute transport during reconstitution. As drug particles dissolve, the solubilized drug concentration increases. The solute diffusion is slow on the order of days compared to the fast dissolution timescales, which are on the order of seconds. There's particle sedimentation, and you can kind of see that in the videos, and this is due to gravity, and this creates a vertical concentration gradient. These simulations indicate that additional mixing, for example, swishing the syringe, would be needed to quickly homogenize the solution on an efficient clinical timeframe. But simulations such as these can help design device usage protocols depending on the formulation. So going a little deeper in these particular simulations, we can actually quantify the dissolution and mixing by plotting the particle size distribution and solution concentration versus time. So the particle size distributions are shown, snapshots of that are shown in the left plot. For a given initial particle size distribution, this would need to be measured for a particular drug product or powder. The dissolution shifts the population to smaller particle sizes over a few seconds. Particle sedimentation under gravity leads to vertical particle density and concentration gradients. And so you can see that in the right plot because there's still a big difference between the min and max solution concentrations spatially. So once the particles have dissolved, there's a vertical concentration gradient, and this would need, if you just left this still, it would take days to mix, potentially days to mix vertically, because this is over about a centimeter. So definitely some active mixing like swishing or some other automated process would be needed to homogenize the solution efficiently. So beyond understanding the timescales for dissolution and mixing, we can use the model to probe the effects of different features of drug product formulations. So one example is, and this is here, we showed that increasing the drug product loading increased both the dilution time, the dissolution time, and drug product solution viscosity. So these types of simulations can complement and expedite formulation development, as well as testing compatibility with reconstitution and injection devices. So I'll turn it back to Sean.
Thanks, Matt. That was really interesting to see how you can use simulations to predict how these drug products are going to form in both the product as well as kind of in a manufacturing environment. So the next example that we're going to talk about is what we call predicting needle coring, which is some work that we published in 2025 with Merck. And you can see some a simulation on the right, as well as a physical test kind of in the center there. This project really highlights the coupling between advanced material testing and finite element simulation that we are known for here at Veryst. The problem in general is that in pre-filled syringes, there's a device called the rigid needle shield, which has both a thermoplastic part as well as a soft elastomer part that the needle then punctures a little bit during storage and transport to protect both the drug product and the needle. You can see the needle and that elastomer there over on the left. But what can happen sometimes is that the needle can experience coring, where the elastomer actually blocks the needle and blocks the drug product from coming out, which is obviously a problem if you're trying to deliver this. Or you can potentially, deliver an elastomer particle into somebody, which is also something that we need to avoid. So we worked together to develop a testing and modeling plan to predict this, and we're going to go through that over the next few slides. So you can see some results from our material testing and modeling and the difficulties that we experienced while we were doing the material testing. So in the upper right, you can see examples of some specimens that we created. These materials were only able to get in their kind of end product form, which you can see in the top image labeled R1 and R2 and R3 are three different materials. These are cylinders that we extracted from the rigid needle shields. So you can see they're about 5 millimeters by 5 millimeters in size. And then over on the right, there are strips that we extracted, again, from those rigid needle shields to get tensile specimens. So there you can see that those strips are maybe 15 to 20 millimeters long and have a width and thickness about 3 to 5 millimeters. So we have pretty small samples that we wanted to test. In the lower left, you can see snapshots of the test that we ran at 4 or 5 different strain levels. So we use a method called digital image correlation during these tests, where we take thousands of images during the test and post-process them afterwards to extract the strain that we apply. And so we get these images, which is a great record. So you can see this huge stretch that these materials undergo before they get to failure, here up to about 500% strain or more. Over on the right, you can see the results from those tests. So there are 5 different color lines. It's a little bit messy there, but you can see the 5 different color lines for the tests that we ran on those 5 different materials. And the solid lines are our experimental data, and then the dashed lines are material model prediction. So we took that data and selected a material model to then use in our simulation, calibrated to fit that experimental data, which you can see there, which you can see in the plot. So these ones here, match very well, and we're very happy with those matches. With the match between the data and the test, we're then able to take that and perform a simulation. So the final simulation that we are running is this needle coring simulation, which you can see on the left. You can see a needle made of metal that is kind of being pushed into a cylinder. This is shown 2D, but this is a 3D simulation. We're just showing that cut face right there. And you can see the colors there are the stress contours in the material itself. And you can see that there's a stress concentration at the tip of the needle, but then you can see that there's a little island of material that gets cut off and cores the needle due to some cutting that's going on in there. And then it sits there. So that's the phenomenon that we are investigating and trying to understand and predict the performance of it. You can see that we're predicting it there. As Matt had mentioned, we try to do model validation for everything that we run. In this case, we were looking at the force versus displacement of that needle insertion test and comparing our model predictions with the actual test there. So again, the solid lines are our experimental data, the dashed lines are our simulation, and you see that we have pretty good agreement between those two. Generally, if the material stiffness and that force versus displacement follows it well, we're catching a lot of features. So if you look at the green one here, you kind of see these humps and dips in the force versus displacement. We see that both in the experimental data and this data, and we see those features kind of repeated throughout. And so we were able to validate that response and really be happy with the way that we were capturing the physics of the material and then the way the needle is inserted in there. We were then able to use this to help to help make predictions on which needles would be best, give the lowest rates of failure, as well as provide some design guidance for future iterations if they wanted to change those. That was a really good segue for us to talk about what we call material modeling or material calibration. So we have a schematic here to show that workflow. Over on the left, we start with our raw materials, and sometimes we're doing tension. That's shown with these dog bones. Those pretty colors that you see on there are the contours from that digital image correlation that I mentioned earlier, or you can see a compression test there. These ones in particular are very small samples because we don't always have a lot of material to work with. The next is kind of the results that we get from that. So you can see some tensile tests in the solid lines on a rate-dependent thermoplastic material, and a dashed line are the material model that we calibrated. So we chose some equations and chose the parameters to match that well, and you can see that we're capturing the rate dependence of this material. We then export that into a material card or an input file for most major finite element codes, including Abacus, ANSYS, LS-DYNA, Altair's, MARC, and COMSOL. And then in the best case, again, we go back and we do a validation. This shows one of our fun validations on a 3D printed lattice structure where we tested the material. That's what that second box is there. We calibrated a model and then we simulated this lattice. And you see very good agreement both between the force and displacement in the bottom, as well as a qualitative agreement between what's happening in those lattice structures as they move through there. Another example of ours is our partnership with Biora Therapeutics, who is developing a smart pill for needle-free oral delivery of drugs into the small intestine. A schematic in one of our simulations is shown over there on the right. The device is extremely interesting itself. It has a miniaturized gas cylinder, which is in yellow there, that is activated by some conditions in your small intestine. It releases gas, which then pushes this piston up, which you see there, which that happens extremely quickly, somewhere on the order of a millisecond. And then the pressure continues to push the piston up. It opens what's called the jet cap, and then there's a drug product in between the jet cap and the piston. There are some holes on this jet cap or the body here, and what happens is you get a jet of material that then shoots out to your small intestine, pierces the small intestine, and delivers a drug in a needle-free way directly to the small intestine. So this can be a nice way to deliver drugs one at the target site that they need. An oral formulation or an external injection isn't necessarily delivering the drug where it's needed. And secondly, you don't have a puncture on the outside of your skin. You're not having to give yourself a needle. This is a pill that you swallow and injects itself, and then you're good to go. So we've been working with them to help reduce their design iterations, improve the design, make a more robust manufactured product, and reduce client time to clinical trials. We've done that in a few ways here. When we started our partnership with Biora, they had some early design and bench tests that they were running. You can see some images there over on the left. So on the upper left, there's an early design, and on the lower left, you can see a bench test where you can see the jet of the material. In this case, I believe, which is this clear right here. So they're testing this and measuring those forces. We were able to quickly sit down with them and provide some value to help them understand how their product was performing and how the different knobs in their device really changed the injection time and the injection force that were really critical to their application. So we were able to do some things with analytical models there and help give them a reduced order model that would give them very good, very strong directional design guidance to then iterate and reduce trials that they needed to run. From there, at that same time, we were building these more higher fidelity activation models to look at the entire performance and get more of those physics in there that we can't do by hand. And now they're moving on to preclinical testing to hopefully get this product out the door and into people's hands and bodies. Another example in combination products is looking at design verification of syringes. Again, one of the things that we like to talk about and emphasize is that accurate models always start with high quality test data. So in this case, we were looking at the response of a stopper and syringe. And again, we extracted samples from the stopper, which you can see in the upper right. So this is the stopper that comes. And we extracted these very tiny specimens, about 3 millimeters in diameter and 3 millimeters tall, and got some material test data and captured that time-dependent response to then model that. We then use this to inform our finite element model of prefilled syringes to make sure that these are going to be safe and applicable for use for patients. One example here is a syringe drop simulation. This is on a thermoplastic syringe. We have also done work on glass syringes. But in this case, we looked at I believe a 1.5 meter drop of that thermoplastic syringe onto a rigid floor and look at how the stress is developed there. And we're really looking for two things. We're looking for the stresses in the body of the syringe to make sure that we're not, going to break this syringe through that impact. But we also want to look at the integrity of the system itself. We don't want the syringe stopper interface to become open and either leak drug product out or allow for contamination from the air into that drug product. So in addition to looking at the strains and stresses in the body of the syringe and the stopper, we look at the interface between those two to understand how that stress state is going to impact the contact there and whether you're going to get any leakage. This can be very nice to help you understand your product performance before you've invested time and money in tooling or prototypes and give you greater confidence that when you perform the tests that the FDA requires, that you're going to pass them.
Sean, should we stop for some questions?
Sure.
I think there's a few. I think they're all for you, actually.
All right, great. Thank you, Matt. Let's see. Ray says, I noticed that the experimental injection time is consistently higher than the simulated time. That one's for you.
I answered that one.
All right. Oh, you answered that one. What was the software used for DIC analysis? So we work mostly with Correlated Solutions VIC products, but I've worked with Trillium's GOM as well as Match ID's software as well, which both the company and the software are called Match ID. So I apologize if I pronounce any names incorrectly. Loic asks, have any of these models been applied for regulatory submissions? I don't know if any of these particular models have been applied, but we have we have experience writing regulatory submissions to accompany your submission to the FDA. In fact, we've had people at our company involved with ASME's VVUQ 40 subcommittee for, oh, I don't know, going on 8 or 10 years or so. So if you aren't familiar, the ASME VVUQ committee is a committee in ASME that has worked to put together a framework for model verification and validation, specifically aimed to help address the FDA concerns when you're using modeling. So we've done both verification and validation of our models for FDA submission. I'd be happy to talk to you about that and kind of the process that requires. I don't think any of these cases here have used that, but it is something that we've done in the past. I think I caught up on all the questions.
There's also, have you tried this on a tissue phantom?
Yeah, I don't know which one this is. If you're talking about the Biora Smart Pill,
No, I think it was before that. I missed it, Sean, when you were
okay.
I think it was probably the I think it may have been the coring.
The coring. No, we didn't do that one on a tissue phantom. I would have liked to, or even on tissues themselves. But in this case, we were most interested in the elastomer clogged. Although tissue coring is also an issue as well because that caused blockage and increased forces.
And on the same topic, what specific approach was used for the needle penetration simulation, ALE, element elimination, etc.?
Yeah, so we were using, we did that in Abacus, which they call CEL, coupled Eulerian-Lagrangian simulation. So it sounds like you're familiar with those types of method. I think each software calls them something slightly different, but they're all basically the same. And the rub there is that, like with these really fine material failure, that's the method you really need to use to get an accurate result. The kind of typical methods, you need such a fine mesh that the computational efficiency goes very down.
And then
goes down greatly.
One last question. I think this is a little more, 7 minutes ago, so more recently. How did you do the strain rate of 200 inverse seconds tensile test?
That's a great question. So we've developed a drop tower here at Veryst Engineering that we've been using for going on 10 years. You might ask, why did you develop one? We weren't really happy with what we could get on the marketplace already, so we developed our own. We have some data, and I'd be happy to share it with you sometime to talk about why how our data looks compared to competitors. Why do we think we can do it better? We're engineers, so we have a lot of hubris, but also our drop tower is more targeted. We focus on testing polymers and polymers only, whereas your big players in the market, your Instrons, your Shimadzus, they make theirs to test everything under the sun, and so they have a broader space that they need to work in, where we're much narrower, so we can really focus on the issues for our materials.
Thanks, Sean.
All right. I think we're all caught up. Thank you all for the questions. Those are great.
Yeah.
And now I'm going to hand it back to Matt.
Great. And keep those questions coming. And if we don't get to them, we'll certainly try to follow up afterwards. So switching now to needle-based injections, we have a great deal of expertise modeling needle-based injections. So this includes intradermal, subcu, intramuscular, and intravitreal. Multiphysics simulations can capture the flow dynamics, chemical transport, and diffusion, and the fluid structure interaction with the surrounding tissue. And these can be anything from kind of the simpler back of the envelope calculations. We did a little bit of that on the dual chamber autoinjector paper looking at different release media, but they can get much more complicated depending on what material properties and some of the setup values that you have. But these simulations can predict the force of injection, hydrodynamic forces on critical areas. For example, in the eye, you would definitely want to know what are the pressures on the retina and the wall shear stress. These models can be coupled to pharmacokinetic models of uptake. And so this is usually a complementary feature to add to development process, but we think it's really useful because there are certain things that certain questions you can ask of models that are much harder to test. And models can also kind of blend together or kind of tease together some disparate experimental values, which may be harder to obtain. And on the subject of injections, we have expertise related to needle clogging. We have deep expertise modeling the non-Newtonian behavior of complex biologics. So these types of things could include shear thickening and thinning, viscoelasticity, yield stresses, dilation, thixotropy, etc. A typical project workflow involves performing the right rheology experiments, then selecting the appropriate rheological model, calibrating the model to data, and then incorporating that into multiphysics simulations. And so on this slide, we're showing results from a continuum model of a dense suspension flow in needles. And this identifies clogging from a specific force threshold. Such simulations can be used to assess injection device performance for a particular formulation, but this could also be used to improve injection device design. So switching gears a little bit more towards medtech, we have experience modeling implantable devices. Certainly, the one I'm going to talk about wasn't meant for drug delivery. Although this is a tissue engineered device. There's a lot of drug delivery research that can be done on these devices. But certainly there are certain injection devices that are being planned as an implantable for really long-term release. But this is a particular example. It's a great example of our industrial partnership with 3D Biolabs, and that's a startup spun out of Mass General Hospital here in Boston. Their scientific founder, Dr. Jay Vacanti, came to us with anatomical drawings and a long-term plan to develop a class of implantable liver devices. And he asked us to design the internal channel networks and support the overall device design using multiphysics simulation and materials engineering. A rough trajectory of our work together is shown on this slide. So the early designs tested the 3D biomaterial printing capabilities and also allowed early surgical procedures to be worked out in small animal models. We characterized the 3D printed biomaterials in-house, and we simulated device loads to provide guidance for robust device design. For example, these devices typically on the order of a centimeter, and they have ports on them. You really don't want any cracking to occur. But the 3D printed biomaterials are very brittle, so it's really important to be careful in the design of things like ports that interface with tubing on the outside. We used computational fluid dynamics simulations to design the inner channel networks to distribute blood or culture media throughout the device. The goal of this was certainly done to nourish the cells within. We developed algorithms to automatically generate the CAD for the complex fractal channels in some of the more complicated devices. Kind of a snapshot, a 3D printed, a picture of the 3D printed version is shown at the right. These were 3D printed and tested in vitro and in vivo. And these devices had distributor and collector networks for the blood or culture media, but they also had these had multiple generations of bifurcations. Some of this work was published in 2024 in PNAS, and a patent was granted last summer. And I will point out that our clients own all the IP generated as part of our projects together, in addition to all work products, including models, test data, results, and design recommendations. Our client work is confidential, so we certainly thank 3D Biolabs and other partners for allowing us to present the material in this presentation.
Matt, there's a question in the chat from Andres. Says, you mentioned PKPD. Is that something that you have previously modeled?
The short is yes. We have one of our colleagues is a chemical engineer, and part of his practice area is applying and combining multiphysics simulations with other kind of lump parameter models like PKPD. So we'd be happy to discuss that more. So just before wrapping up, I'd also like to point out that we do a wide range of work in areas complementary to combination products, including upstream processes, including biofilling and drug product lyophilization. Multiphysics simulations that are shown here can be used to help develop operating protocols and troubleshoot issues. For vial filling, issues include splashing, jet breakup, and air entrainment, and their dependence on flow rate ramping, the nozzle motion, its distance from the liquid, and the drug product viscosity. For lyophilization, issues include what's termed vial fogging. This is where there's a residue that can remain on the vial walls, and other issues associated with producing a powder or a lyo cake. So with that, we will conclude this presentation, and we will shortly move to Q&A. We certainly thank you for your attention. You will receive a link to the recorded webinar next week. We also kindly ask that you fill out our 2-question survey via the link or the QR code. If you'd like to see additional content on combination products in future versions of this webinar, please let us know. Or if you'd like us to cover other topics in our webinar series, please reach out to us. You can certainly find our future webinars on our website, and we'd now be happy to answer your questions.
Yeah, thank you all for attending and all the great and thoughtful questions. And as Matt mentioned, I want to thank our partners who allowed us to share this data, because it's not often, or share these projects, because it's not often that we get to do that. I was trying to keep up there. So I think there's the latest question that I think we need a little more information on, that just says, silicone spraying pattern. Not sure if you're asking if we can model that, or if you're talking about silicone sprays that get applied to prefilled syringes or other combination products, but happy to talk about that.
Oh, here's some more in the Q&A. Yeah, and this might refer to the lubricating layer, but sure, we'd certainly be happy to answer those questions. We don't just do, I mean, my area is microfluidics, and then our colleague also does even smaller scale things such as nanofluidics, and there's just a lot. It's a different world. It's not more complicated. It's just different forces dominate. Certainly at the sub-millimeter scale, surface tension dominates, surface forces dominate, electrostatic forces. But you go smaller than that, down to the nanoscale, you can get things like van der Waals force that matter. So we're happy to talk about that if you think your problem may involve that. I see a number of questions. Are these models typically handed off to the clients, or does Veryst Engineering continue to maintain the rights to use LCM? Oh, great question. So again, our clients own our work. It's confidential. The odd time we may develop an internal capability. Sean mentioned the drop tower. We certainly, know all kinds of tricks in the simulation packages we use. There's certain frameworks, but anything that is involved with the client confidential data, that's sealed off. The client owns it. The other thing is we get such a wide range of problems. It's kind of, sometimes we can reuse some of the tricks that we find in the software. But it's one of the nice things about being in consulting is just the wide variety. But again, we always hand over all data, methods, IP, and simulation models to our clients.
Yeah, and so someone had asked, and I forget who, so I apologize. How do you choose the software? The answer I put there was, we'll choose the software that we think is best from the kind of that list of software that we use. The other answer though is sometimes the client says, we have people who can use Abacus or ComSolve, so that's where we want the model in, and we do that if we think it's possible. You know, there are problems that we can solve in ComSolve that we can't solve in Abacus, and vice versa. So sometimes we can't do that request, but we will as much as we can. And you know, the models are our clients. So sometimes we develop a model and then it gets handed off to the client and they use it for months or years or however long.
There's a question about links to the published work. We can certainly provide that. And then there's one, be interested in hearing more about intravitreal injection simulation, any published material. So we don't have any published material on that, except we do have case studies on our website. So you can read more. There's a couple. And we don't have any, say, peer-reviewed publication. The thing is that much of this work is related to clients. So we make a case study, it has to steer clear of any of the anything related to confidentiality. But it's interesting because, you know, there's certainly a lot in the literature, different injection methods, what's important, and depending on what the release media is, for anything around the eye, you have to be very careful about the flow within the vitreous. And the vitreous is kind of a complex fluid. You know, viscosity varies, but it's kind of a somewhat like a gel, and the concentration may vary spatially. But one has to be really, really careful that the jets, due to the injection, don't hit the retina. Or if they do, you really have to keep that pressure low. And that's something that simulation can really be helpful with. And then you add, you know, because there is a difference as for a given needle size and a different volume that you're going to inject, controlling that injection profile is really important. I looked in the literature and it's actually pretty hard to find an injection time when you're talking about intravitreal injections. What you'll typically see, if it's set at all, is slow and steady. That's just not very precise. So there may be an opportunity there to, you know, do some automation and control the flow rate better and do some A/B tests. You know, is a 3-second injection time for, say, 40 microliters better than 1 second? So happy to talk more. Oh, have you looked deeper into the microscopic multi-scale models to simulate stopper barrel friction? The short answer is yes. We don't have anything that we can just point you to, but there is a lot going on, and that's a nice fluid structure interaction problem because the stopper is overfit to the barrel. It pushes up against the barrel, that thin layer of lubricating liquid. There is a squeeze film concept, which is kind of a classic fluid mechanics concept where the thickness of a layer of fluid gets smaller and smaller if you're pushing, say, a solid down on another solid, and there's a layer between a fluid between them. Maybe it's air, maybe it's liquid. But as it gets thinner and thinner, the flow resistance goes up considerably. And because flow resistance scales as the thickness cubed in this case. So basically, so that's one of the things that makes it interesting, but then also the van der Waals forces also start to come into play, especially around the 100 nanometer and below mark. So we certainly have the backgrounds collectively to address this problem. So if that's of interest, happy to talk to you. That's why we measured in the lab. It was just easier, and we surprisingly got that power law curve, because if you look in some of the literature, most people just fit a line to the larger speeds and they fit that line all the way back to 0 speed, but actually that's not what happens. It actually, that force of resistance or the stopper friction it goes to 0, but it is a power law as you decrease the speed.
So I think we've addressed all the questions there. If we've missed any, please let us know. But again, thank you for attending. We would really appreciate if you took our little, two question survey there over on the left. Gives us great feedback. And if you want to reach out for additional follow-up or any specific questions that we didn't get to, we'd love to talk to you and share more about what we can on all these projects and our capabilities. Anything else you want to add, Matt?
No, just thank everyone for attending, and the questions have been great. We've really enjoyed this.
Yeah, I think that's my favorite part of these things is when people ask great questions, because that's where I feel it's the most interesting. All right. Thank you all very much. You'll be getting a recording probably early next week of this webinar in your email inbox. And we'll try to add some other information there about kind of the links to the papers that people have been asking about. But feel free to reach out to us if you don't receive that. We'd be happy to chat. Bye.